Today, in the field of repair and restoration of damaged tissue, I will talk to you about regeneration. What would be best as a repair if we could just simply restore from our stem cells an intact tissue or even an organ. And in this field, you know that amphibians are like the king. You can So in the newt, for instance, you can section the limb and it regenerates. In xenopus frog, you can section the tail and it regenerates. So they have this wonderful power of regeneration. And in our lab, we are trying to understand the mechanism underlying this regenerative capacity with the aim of potentially triggering this in patients. And our model is the retina. So as we've heard this morning, there are different types of retinal disease. I will today focus more on the degeneration of photoreceptors. And in this case, you probably know the age-related macular degeneration. In France, it's DMLA, AMD, or genetic disease like retinitis pigmentosa, which leads to photoreceptor cell death. And as there's no spontaneous regeneration, this leads to vision loss. In the worst case, to blindness. Depending on whether it's a genetic disease or not, depending on the stage of the disease, there are obviously different types of potential therapies. So today there are, I would say, almost no cure for these diseases, but there are many different approaches. You've heard already, and you'll hear probably from Denise Dalcara about gene therapy, gene augmentation, which can be done at early stage of the disease. But at later stage, when photoreceptors are already gone, you've heard about transplants, about optogenetic therapy, but today I'd like to tell you about cellular therapy. And not the one where you manipulate stem cells in vivo and do transplantation, but whether we could self-repair our own retina from our own stem cells. So in this matter, I need to introduce you with one important cell type in the retina, which is of interest for regeneration, which is a glial cell, and which is called the Mueller cell, this purple one here that spans the whole thickness of the retina. And these Mueller cells in the fish have been shown to be able, in case of injury, to re-enter into the cell cycle and regenerate all types of damaged neurons. As I told you, this is not the case in the mouse, in mammals. Upon injury, these cells re-enter into a gliotic state, but they do not reprogram into stem cells, and so they do not proliferate. However, and this makes the whole difference and the whole interest in this subject, is that it has been shown a few years ago that you could, if you add some growth factors or overexpress some transcription factor, these cells can exit quiescence and proliferate upon injury and regenerate sometimes some types of neurons. This remains, however, highly inefficient, and it's clearly not enough for repair. But it shows that these Mueller cells, even in mammals, have retained some stemness capacity. And we think that if we learn more about how it happens, how the mechanism underlying this mechanism in species where it spontaneously arises, then we could maybe better figure out the key determinants that could trigger this regenerative capacity in mammals. And so with this in mind, finding species which have this spontaneous ability, we were interested, as I told you, amphibians are really good at regeneration, and we wondered whether amphibians could be also good models to study the mechanism underlying Mueller cell regeneration. And so we developed these last few years different models of retinal injury in an amphibian species, the xenopus frog. Either we perform simple mechanical injury, so we do just a needle poke injury, and then we can analyze the impact on Mueller cell around this injury. or we develop a transgenic line allowing for conditional and reversible photoreceptor ablation, here cone rod cell ablation, or we also generate a CRISPR model targeting the rhodopsin gene, which is known to be highly frequently mutated in patients afflicted with retinitis pigmentosa. And in all these different injury models, we looked at the behavior of Müller cells, and what we found is that indeed, in amphibian, in the xenopus tadpole, Müller cells, upon this injury, are able to exit quiescence, proliferate, and regenerate photoreceptors. So that makes xenopus an additional model to study retinal regeneration. And so I'd like now to present you how we could, using this model, identify key determinants for regeneration and use this knowledge to trigger regeneration in the mouse. So we first focus on one signaling pathway, the hippo-YAP pathway, which is a signaling that has been shown to be very important for cell proliferation and stemness behavior, and it regulates the organ growth during development. And just what you need to know is that on this signaling pathway, we will focus here on the downstream effector of the pathway, this YAP construct, this YAP factor, which is, upon the activation of the pathway, is phosphorylated and which makes it inactive. So it remains in the cytoplasm or it's degraded. And it's when the pathway is off that this factor can enter into the nucleus and activate as a co-transcription factor the target genes, right? And so we found actually that this factor, YAP, is specifically expressed in Müller cells. You see with a double staining with a Müller cell marker, there is a perfect match. And originally we were quite surprised to see this expression in quiescent cells because these Müller cells do not proliferate. So we wondered whether this factor could actually confer some stemness potential in these Müller cells in case of injury. So to address this question, we do what we usually do in our field. We try to eliminate this gene and look at the impact on the regenerative capacity of the retina. And so we took advantage here of a transgenic line that allows for conditional expression of a dominant negative form of YAP. And so we combine this transgenic line with a retinal injury, and we analyzed Müller cell response to this injury. So here in a control situation where you can see here in green, there's the nice layer of the photoreceptors. You can see at the site of the injury, there are plenty of Müller cells that re-enter into the cell cycle. You can see them in red here. But when we interfere with YAP function, then at the site of the injury that you can see here, there's no cell cycle re-entry of these Müller cells. So this tells us that this factor seems to be absolutely necessary for these Müller cells to re-enter into the cell cycle upon injury. So we thought, can we use this factor to awake dormant Müller cells in a mouse retina? And so that's what we did here with, so how do we proceed? We use retinal explants that we culture in the presence of an AAV that overexpressed this YAP construct. What I have to tell you here is this AAV, it's this SHH10, which specifically target Müller cells. So we can overexpress YAP specifically in Müller cells. And we use a construct that is mutated so that it cannot be phosphorylated by the hippo pathway. So it's a constitutively active form of YAP, okay? And in this context, we can see that in contrast to a control situation where Müller cells in blue here are mainly not proliferating, they do proliferating very strongly when we overexpress this YAP. So you have simply to overexpress one factor to make these cells proliferate as if they were in a xenopus retina. You have close to 80% of these Müller cells that do proliferate actively in this degenerative condition. So this was ex vivo in this retinal explant model. Now I have to tell you that this was confirmed in vivo using intravitreal injection of this AAV in this case, you still have many Muller cells that do re-enter into the cell cycle. So YAP seems to be a potent inducer of Muller cell reactivation. So here I've told you so far that we've shown that xenopus is a model where Muller cells do re-enter into the cell cycle and regenerate photoreceptors, and that this requires YAP, since when we remove YAP, Muller cells remain quiescent. And on the other hand, in the mouse retina, while normally these Muller cells are quiescent, you simply have to overexpress one factor, YAP, to allow them to re-enter into the cell cycle and proliferate. So that was an example of an intrinsic factor that we determine, that we identify in xenopus, and that could help to trigger Muller cell cell cycle exit in a mammalian retina. Now, I'd like to show you in the next part of my talk that intrinsic factors are clearly not enough, and that the microenvironment, the niche in which Muller cells reside, is obviously highly important. And this started with a comparative analysis where we initially wanted to compare Muller cell response to injury in two different xenopus species and during the aging of the tadpole. So at the time, I don't know if you have to guess, but at the time I was guessing that in two xenopus species, so closely related species, Muller cells would respond similarly, right? And along the while the tadpole ages, I have to tell you, in the mouse, I told you that you can trigger some reactivation, but this is quite efficient in a young mice, right, in a very early postnatal mice. But the older the mice is, the less efficient this will be, right? So here, if I had to guess, I would say, I would have said that Muller cell would respond similarly in the two xenopus species, and that this capacity would decrease while the animal is aging. Now, I have to tell you that I lose all my bets, so you'll see that this was also the case here. So to compare two different species, we obviously have to design the exact same paradigm of injury. So here we designed this CRISPR model targeting the rhodopsin gene in the two species, right? So that's what we did, and when we compare the efficiency, the models are extremely similar. Photoreceptors that you can see nicely here in green in Tropicalis and levis are almost completely gone in the two models. So very efficient degeneration, very good model mimicking the retinitis pigmentosa disease. Now, when we look at Muller cell response to injury, while in Xenopus levis, I told you already, these cells do respond to the injury by re-entering into the cell cycle. You see plenty of them proliferating. This was not the case in Tropicalis. So although these species are really closely related, right, there is something that differs between them, so that in levis, it uses mainly its Muller cells in response to injury. This is not the case in Tropicalis, but instead, it uses another type of stem cell that is localized in the periphery in response to this injured retina. Now, not only we found this difference between these two species, but even during aging in levis. So when I told you earlier that, yes, it's working in levis, Muller cells do respond to the injury by proliferating, this is actually only true in very late tadpole stage. If you look at earlier stages, these cells very few do respond to the injury by re-entering into the cell cycle. So completely counterintuitive with what we could have imagined about the aging of a Muller cells, right? You would have thought that a young Muller cell would be more plastic and re-enter more likely into the cell cycle, right? But this was not the case. But we considered that this was a fantastic model, right? In a single species, you have a stage where it works, a stage where it doesn't work. What makes the difference? We could compare by doing transcriptomic, compare what's the difference. And so that's what we did. In our model of retinitis pigmentosa, we looked deregulated gene upon the degeneration at a I forgot to tell you that this young stage, I will call them refractory, since they don't respond well, and the late stage, permissive stage. And so at both stages, we look at deregulated genes, and we compare the two. And what we found by bulk RNA-seq analysis is that obviously in the two stages, a lot of deregulated genes are linked to light stimuli, light detection, because it's a degenerative model where photoreceptors degenerate. But what we found quite interesting is that only at the permissive stage, we found all these genes that are upregulated and that are linked to the immune response. And they are upregulated only at the permissive stage and much less at the refractory stage. And here is an example of a set of genes that you can show here very easily, that all these genes at the permissive stage are strongly upregulated upon injury, in red here, and much less here at a refractory stage. And among all these genes, there are many genes linked to inflammation and markers of microglia. So the next step was to wonder what was the difference in microglia between these young and these late stages. So first, we just look at the presence of these microglia in the retina. And again, we were quite surprised to see that microglia in these young stages, which is quantified here, are almost not present. There's almost no or very few microglia in this young tadpole stage, and this number starts to raise while the tadpole is aging. So it seems that at the refractory stage, microglia have not yet colonized the retina, and that was quite intriguing to us because in other species, we know that microglia infiltration starts already during development. So then we went earlier during development to know what's happening there. And so here we took advantage of a transgenic line, allowing us to monitor microglia, to quantify the number of microglia infiltrating the eye. And what we found is that actually, yes, there are microglia entering into the retina during development, but then this number drops at the end of embryogenesis, reaching almost zero at the end of, in the early stage of the tadpole. So if we pull what I've told you during the tadpole stage and what we found here in embryonic stage, it seems that there are two waves of microglia colonization in the retina, one during the embryo and one at the tadpole stage. That leaves us with a window during the early tadpole stage, so perfectly when the tadpole is refractory, with a quasi-absence of microglia within the retina. So obviously we wondered whether this could be linked to the capacity of regeneration. So we tested first what happens in case of injury, because this is during physiological condition, right? So upon injury, what we found is that, as I said, in a control condition at this early stage, at this refractory stage, there are almost no microglia. Upon injury, there is a little increase, but very little compared to what happens at the permissive stage. You see plenty of activated microglia there. So it seems that the more microglia in the retina, the better Müller cell will proliferate. Although this is only correlative, to know whether indeed this is causal and whether this could explain why in a young tadpole Müller cells do not regenerate, we simply wondered whether could we awake this refractory Müller cell by simply generating an inflammatory context, right? So to do that, I have to introduce with another model of retinal injury, which we trigger by cobalt chloride intraocular injection. This is quite toxic and trigger photoreceptor degeneration, and in particular of cones. cones are almost completely gone. Remember that for afterwards. And rods, little bit also, but mostly cones are affected. Now, why are we interested in this degenerative model compared to the previous one I told you? It's because in addition to triggering this retinal degeneration, it also triggers a fantastic inflammatory environment. You see on a flat-mounted retina, here in white the microglia, you can see how strong the increase is. Okay, you can tell me that, but in any retinal disease model, you will have an inflammation. This is correct, but in comparison, this COCL2 model triggers much more increase in microglia, and you can see how activated they have. They have this amoeboid shape, while in our rhodopsin knockout, microglia are much less numerous and have this ramified morphology. So then we have a nice model triggering retinal degeneration and an inflammatory context. So could this, in this condition, could this be sufficient to awake refractory Müller cells? So when we did this experiment, indeed, we managed to see many Müller cells that in this case exit quiescence and proliferate. So that doesn't demonstrate for sure that it is due to inflammation. So to make sure that it was associated to inflammation, we repeated this experiment, but this time by removing microglia in this COCL2 degenerative condition. And in this case, the number of Müller cells decreased, suggesting that this upregulation was really due to the inflammatory context triggered by COCL2, right? So what I've told you here is that in a young tadpole, there is very little microglia, so very little inflammation, and we propose that this is the reason why Müller cells are refractory. And on the other hand, in an old tadpole, microglia have infiltrated the retina and allows to generate an inflammatory microenvironment that favors Müller cell cycle reentry. And we have shown that by a simple immune challenge, we can confer to this quiescent cell proliferative activity, suggesting that they were not refractory because of intrinsic barrier, but really that it was the microenvironment that was key to determine whether Müller cells can proliferate or not. And so we wondered whether could, from this knowledge, make other dormant Müller cells in other species, can we awake them by simply generating an immune challenge? So I told you, remember, in Tropicalis, this was Müller cells were refractory, and same thing, can we also awake Müller cells in mammals using this inflammatory microenvironment? So first in Tropicalis, we did this COCL2 intraocular injection. I told you this generates an inflammatory context. Now, could Tropicalis, in this case, answer positively? The answer is yes. Now we see it proliferating, as in Levis. So by simply changing the microenvironment, these Tropicalis refractory Müller cells now proliferate actively. In the mouse, how do we proceed? Again, this retinal explant paradigm, where this time to trigger inflammation, we used LPS. And in this case, again, what we saw, that even in mammals, by simply generating activated microglia, then Müller cells do proliferate significantly. So this shows that the microenvironment is sufficient to trigger Müller cell proliferation. Now, is it absolutely necessary? And to ask this question in the mouse, because normally they do not proliferate, we have to trigger their proliferation and see whether these microglia are necessary for this proliferation. Okay, I hope you still follow me. And to trigger Müller cell proliferation, we go for our intrinsic factor I told you about, this YAP factor. Remember how a potent inducer it was. When we use this AAV YAP, Müller cells do proliferate like crazy, okay? And remember, it's a constitutively active form of YAP, so very potent inducer of YAP. So again here, if I had to bet, I would think that even in absence of microglia, even in absence of an inflammatory context, if you overexpress intrinsically in Müller cells this YAP construct, you would allow them to re-enter into the cell cycle. But actually not. So when we did this overexpression of YAP, but in absence of microglia, then you lose this strong mitogenic activity, suggesting that microglia are absolutely required for these Müller cells to re-enter into the cell cycle, even if you overexpress a mitogenic factor like YAP. Now, okay, it seems that inflammation is absolutely fine for triggering Müller cell proliferation, but regeneration is not only cell cycle reentry, right? Now, then, when Müller cells do proliferate, they have to undergo neurogenesis and regenerate neurons, right? So we wondered whether in this favorable context of proliferation, is it also associated with a favorable neurogenic activity? So to look at this, we use again this COCL2 model, at a permissive stage. So when Müller cells do already respond to the injury in any type of injury, but when we use the COCL2 model, then it's almost 100% of Müller cells that do proliferate. So really they love to proliferate, they love this inflammatory context. But when we looked at neurogenesis, so this time we did exactly this experiment, so where almost 100% of Müller cells do proliferate, so we could have expected a very effective regeneration, but when we look 1 month later, and we look at cone regeneration, you see, remember, cones were the most affected cells in this model. Cones are still not repaired at all. So very bad condition for neurogenesis, actually. Now, are these cells not doing any neuron at all? Using a Pearl Chase analysis, we looked at different cell type, and we found that here, again, the cones are not regenerated at all, but still, these proliferative Müller cells can make some types of neurons, but not biased towards the cell type that is lost, which is a pity for regeneration. You want to do the neurons that is degenerating, not the other ones, right? So now, what about in the mouse? We saw also in the mouse that triggering an inflammatory context helps Müller cells to proliferate. What about their neurogenic potential? So here we did this experiment of LPS treatment, which triggers inflammation on the retinal explant. And then we look at neurogenic gene, like ASCL1. And this gene is actually not at all upregulated. It's actually rather downregulated. So again, good for proliferation, bad for neurogenesis. But then we thought, how can we could maybe help the neurogenic effect by removing inflammation. But if inflammation is absolutely required, I told you it's absolutely required for the first step of the regeneration, how can we manage to have this inflammatory context first and then the inhibition of inflammation? So to address this challenge, we thought we have to have a dynamic inflammatory context. And for that, we did sequential treatment. First, we trigger an inflammatory context with the idea of triggering Müller cell quiescent exit. And so the first step of regeneration. And then ablate microglia to allow inflammatory resolution and then allow potentially neurogenesis to occur. And in this case, indeed, we found that this sequential treatment allows Müller cell proliferation and this significant upregulation of neurogenic gene. So as a conclusion, although in the field, it's rather like the dogma is that inflammation in mammals is rather detrimental for regeneration. And indeed, we saw that neurogenesis is affected by, is negatively affected by inflammation. However, our data suggests that although it may inhibit neurogenesis, it is activated microglia are absolutely required for the first step of Müller cell reactivation. And so we propose here that this effect is really conserved among vertebrates and not as suggested in the literature where they could have different effects in regenerative versus non-regenerative species. Here we think that when we sequence, when we look at the different sequence of regeneration, actually inflammation has similar pro-mitogenic effect on Müller cells, both in xenopus and in the mouse. But since it has this deleterious effect for the neurogenic step, we believe that we have to play with the environment to make it friendly for regeneration, first for proliferation, and then for regeneration by, in the second step, inhibiting this inflammatory context. And so with this schema, I'd like to finish by thanking all the people in the lab involved in this work, and in particular for the xenopus part, it's mainly Karine, Alicia, Albert, Léa, Jinxian, Caroline. And for the mouse part, it's Sophie, Demi, Jérôme, Juliette, and Patrick. And our collaborators, also Jan Odik from Rennes, who help us with the xenopus sequence analysis, and Denise Dalcara for the AAV the AAV YAP that I mentioned. So, and thank you very much for your attention.